All right, everybody, thank you again for coming. This is Mr. Zawacki, and today what we're going to be talking about are three things: DNA replication, transcription, and translation. So those are the three things that we're going to be discussing today, both working for biology and also anatomy. For biology, this is lesson 305. For anatomy, this is the lesson where you learn about DNA and the things it can do. All right, so first of all, this is the easy one, DNA replication. All replication means is just like to copy, to make an exact copy. And that's what DNA replication means. It's where DNA makes a copy of itself. It is done during the S phase of the cell cycle. Remember how we have the cell cycle and you have most of it being interphase? And then you got mitosis and cytokinesis is the other parts of the cell cycle. Well, the S phase is one part of interphase. So this is where DNA makes a copy of itself. The original DNA strand, first of all, is unzipped by this enzyme called helicase. That's something you definitely want to know. DNA polymerase is the next step, and all it does is base pair. So the first step, you unzip the DNA like a zipper. Second step, DNA polymerase, another enzyme, base pairs everything together. And the base pairing is very easy. A will always go with T for DNA. T will always go with A. So these kind of are all together. G will always go with C. C will always go with G. So it's pretty simple for base pairing for DNA. If it's a T, it matches up with an A. If it's an A, it matches up with a T. And then G and C are the same thing as well. The two strands are either leading or lagging. Remember how DNA unzips itself? Well, there's two strands. There's a lagging strand and a leading strand. The leading strand, it's done in one giant piece. A to T, T to A, G to C, C to G, just all the way through. One straight piece. The lagging strand, though, is in many pieces. So think of lagging, like you're falling behind or you're not as going as quick as the leading strand. Well, that kind of makes sense because the lagging strand is done in pieces. And those pieces are called Okazaki fragments. And then the last step, those Okazaki fragments, they're glued together by ligase. Final product, 2 DNA strands from 1 DNA strand. So going back to this, I'm going to explain it one more time with a picture. These are the things you need to know. You need to know what helicase is. That's the thing that unzips the DNA. You need to know what polymerase is. That's another enzyme that base pairs everything. You need to know what Okazaki fragments are. Those are the pieces on the lagging strand. And you need to know what ligase is. Ligase is glue, kind of. It's another enzyme that glues together the Okazaki fragments. So here it is again. So the dark blue is the original DNA, the parental DNA. Step 1, helicase. Remember helicase? That's the thing, the enzyme that unzips it. So it's like a zipper, unzipping. Step 2, now we have step 2 and step 3. Now we have DNA polymerase base pairing. So here's DNA polymerase. If it's on the leading strand, it's done in one giant piece. If it's on the lagging strand, 3 to 5, it's done in smaller pieces. And those pieces are called Okazaki fragments. Here's one. Here's one. Here's one. And remember, they're glued together by ligase. So that's what you need to know. Helicase unzips it. That's the enzyme that does that. DNA polymerase base pairs everything. If it's straight through, it's the leading strand. If it's in pieces, called Okazaki fragments, that's the lagging strand. And those lagging strand pieces, called Okazaki fragments, they're glued together by ligase. Final product, 2 new strands of DNA. So make sure you guys know those. I'll ask you questions about them at the end. All right, and then the other parts, transcription and translation. How I want you to think of this, guys, is part 1 and then part 2, because they go together, but you got to have part 1 first. Part 1 is transcription. This is done in the nucleus of the cell. Remember the brain or control center? It's where the DNA is found, so that makes sense. And what happens is, just like DNA replication, remember how helicase unzips it? Well, instead of making another copy of DNA, it actually makes messenger RNA. That's the product. That's the product of transcription. Think of this as a code. So the point of transcription, guys, is for DNA to make a code called messenger RNA. It happens in the nucleus, and it happens pretty similar to DNA replication. Helicase unzips it, but instead of DNA polymerase base pairing everything, RNA polymerase does the base pairing. And it's pretty similar. G goes with C, that's the same. C goes with G, that's the same. Here's the difference, though. Sorry, this should be a T here. T goes with A. That is also the same. Here's your difference. A does not go with T anymore for RNA. A pairs with U, uracil. It's a new nucleotide piece, but it is only found in RNA. That's why there's that difference. DNA, if this was DNA polymerase doing this, it would be everything the same except A to T. If RNA polymerase is doing it, there's one change. Instead of A to T, it's A to U. So instead of, yes, Joshua, good job. So instead of thymine, RNA pairs with uracil instead of thymine. That's your only difference between DNA polymerase and RNA polymerase. Yes, and it would change the chemical makeup, because remember, you're not making the same thing. So DNA replication with DNA polymerase just makes 2 copies of DNA. But with RNA polymerase, it's going to make messenger RNA, which is a completely different code. So that's part 1, make the code, RNA, messenger RNA. Part 2 is translation. So now, after you get part 1, after you get this messenger RNA code, now this is done in the cytoplasm, so all the extra space inside the cell, and the mRNA is translated by a ribosome. So a ribosome helps do this. We know what translate means, just means to read something and decode it. Well, a ribosome does that, and it reads each part of the mRNA 3 letters at a time. That's called a codon. It's another vocabulary word. 3 letters of a messenger RNA is called a codon. Now the tRNA, which is another character in the game, this one just transfers. Think of T for transfer. All it does is read the code and then transfers the amino acid that the code is for. And then the amino acids are linked together, creating a protein. This, right here, the protein, guys, is the final product of transcription and translation. So to put it all together, think of it like part 1 and part 2. Transcription starts with DNA, but it uses RNA polymerase to make messenger RNA. Part 2 translation, that's where the messenger RNA gets translated 3 letters at a time by a ribosome. And then the ribosome says, okay, tRNA, bring over this amino acid. And then it does. And then you get all these amino acids linked together, like peptide bonds, and that creates a protein. So the entire point of transcription and translation, guys, is to make a protein. The entire point of DNA replication is just to make another strand of DNA. So there's your big difference there. Here is transcription. Remember, this is part 1. So we have DNA up top, but look, G to C, C to G, A to U, T to A, G to C, G to C, A to U, C to G, G to C, T to A. Very simple. So here's the product of transcription, mRNA. So that's easy. All it does is RNA polymerase just base pairs a piece of DNA to create messenger RNA. Translation, remember, here's your messenger RNA right here. The messenger RNA now needs to get translated. This is part 2. But once you get the messenger RNA, which is right here, the blue line, now the ribosome, this big purple thing, is going to read each letters 3 at a time. C-U-U, C-A-C, G-G-U, C-G-U, U-A-C, A-G-G, and every time those 3 letters are read, an amino acid is placed that matches up perfectly. Here's another amino acid, perfect match. Here's another amino acid, perfect match. Here's another amino acid, perfect match. So what I'm trying to tell you guys is, for translation, the messenger RNA code gets translated. The ribosome translates it. The tRNA transfers the amino acids over. And then you just have all these amino acids that are grouped together. And then finally, you have proteins. So that's it. You're going to create a primary structure first. This is the most basic structure of a protein. If the structure gets more complicated or more complex, it can become a secondary structure, like a helix or a pleated sheet. So this is all secondary. Tertiary structures just look like a glob. They're even more complex. And then quaternary structures are several globular structures put together. So main thing you guys need to know is the products of transcription and translation. So do we have to remember all the amino acids that pair with the codons? No, you don't. There's actually a table that they'll give you, so you don't need to memorize any of the amino acids. You just need to be able to read and understand the table and see how it works. If you guys did the assignment from 305, you'll know what I'm talking about. So we're going to do a little bit of a practice now. Speaking of that table, this table that Joshua asked about is right here. How to read it, look, you look at the first letter, second letter, first letter, second letter, third letter. Or you can just find it in the charts here. So the first thing we're going to do together is look at this, transcription and translation activity. So with the DNA code is right here. The mRNA sequence is right below. This is very simple. Remember, G pairs with C. T is with A. A is with U, and I'm going to draw a line. T with A, C with G, and another C with another G. There's another line there. So what I want you guys to do is do the first 5 sets. So I already did 2 for you. So I want you to do the first 5. So I want you to break down these 3, and these 3. I want you to come up with the mRNA sequence for all of those. But then the tricky part, I want you to look down here. Look, it's CAU. I'm going to look down here and look for CAU, and I found it. It's right here. CAU is HIS. So that amino acid is called histidine or histidine. So that's what I want you guys to do. So your answer will be 5 amino acids in a row. I don't really care about the mRNA sequence, because if you did that right, you'll get the amino acids right. So what I want you guys to do, once you're done, is tell me the 5 amino acids. So it should be HIS, comma, whatever AGG is, whatever this one, and this one is. So once you take about a minute, and you should give me 5 answers, back to back to back to back to back, all together in the same message, starting with the amino acid HIS. So I'll give you guys about 1 or 2 minutes to do that, and we're going to do it one more time after that. Good job, Marcus. Looks like you got those right. Just checking over people. Remember, you should be giving me 5 amino acids for your answer. I don't really need to know the mRNA sequence, because you're going to need to use that to come up with the amino acids. So HIS would be first. We just did that together. If I look at AGG down on the bottom, AGG stands for ARG. So now I got ARG. AUU, if I look at AUU on the bottom, where is it? AUU is right here. So AUU is going to be that amino acid, LU. CUG, if you look for CUG, you'll see it's LEU. And then AAG is LYS. So does everybody see how we got those answers? All you have to do, guys, the first step, and this is just like your assignment from 305. The first step, you just have to figure out the mRNA sequence. C to C, T to A, A to U, T to A, C to G, C to G, C to A, T to U, T to U. So that's the first thing. The second thing is you have to use the amino acid sequence chart. You just have to find it and see which one it is. And this one down here looks like H, a little bit hard to read, but it looks like ILE or something like that. So anyways, you'll use the amino acid sequence next to figure it out. That's how to do it. So next thing, I want you to do it one more time. I want you to do the first 5 sets. This ACC, CTT, CAT, GGG, CCC. I want you to do that one more time, but I want you to tell me these. Tell me these 5 amino acids that go with that one. So I'll give you guys another 1 to 2 minutes to do that. Give you guys about another minute or 2. I just did the mRNA sequence for you guys. It should be pretty simple. Remember, you're just base pairing. UGG is the first amino acid, and it should be TRP, troponin. So I want you guys just to finish the last 4 and tell me the 5 amino acids that should be coded with the second bottom strand. So once you get those 5 amino acids, just type them, just one after, all 5 in a row. All right, so just looking over everybody's, it should be TRP, GLU, VAL, PRO, proline, and then glycine, GLY. So those of you who got that, Joshua, Bailey, Francesca, Taylor, yeah, Emma, Ciara, Madison, Justice, good, Vincent. Very good. So guys, that's it. That's how to do transcription and translation. That's how to do your assignment for 305 for biology. So that's how to do that. So make sure you guys understand the point of that too. The point of that is to then group those amino acids together to form a protein. So that's the whole point of it. Now, a couple follow-up questions, though, just to make sure we know our enzymes that help out. Can anybody type the name of the enzyme that unzips the DNA to get everything started in both replication, transcription, translation? What's the name of the enzyme that unzips the DNA? Good job, Madison, Joshua, Francesca, Ciara. Yep, you guys got it. Helicase. Helicase unzips everything. That's the enzyme. Now, we know one is DNA and one is RNA, but what's the name of the enzyme that base pairs? What's the name of the enzyme that does the base pairing? Anybody remember the name of the enzyme that does the base pairing? Starts with the P. Yep, Madison, you got it. DNA polymerase base pairs for DNA. RNA polymerase base pairs for RNA. Very similar, except for RNA does A to U. Good job. Does anybody remember the leading strand is done in one giant piece. The lagging strand, though, on DNA is done in several pieces. Do you know what the names of those pieces are called? Definitely a weird name, probably developed by the guy with the same last name. Does anybody remember what those pieces are called? No, close. It's in here. Remember the pieces of the lagging strand. There you go. Remember Okazaki fragments. If they're fragments, they're pieces, right? So the pieces are called Okazaki fragments. And does anybody remember the name of the enzyme that glues the fragments together? Good job. You guys got it. There you go. So that's the main stuff you guys need to know with replication. Remember, helicase unzips the DNA. Polymerase base pairs everything. The pieces, remember, if it's in one strand, it's leading. If it's in pieces, called Okazaki fragments, that's on the lagging strand. And then ligase is the glue that glues the pieces together. So that's all you need to know for replication. Remember transcription and translation, guys. Transcription is just, the point is to make a code. And it's done by RNA polymerase. And it happens in the nucleus. And then translation, the point of that is to make a protein at the end. So you use the code and you translate it 3 letters at a time, and each 3 letters stands for an amino acid. And then finally, those amino acids are grouped together to form a protein. Here's transcription again. Remember, RNA polymerase, base pairs to make mRNA. So now you got your code, and then mRNA is translated 3 letters at a time, called a codon. And the ribosome translates it. And the tRNA transfers the stuff. And the tRNA will transfer the amino acids that match up with the specific codon. And then you have a group of amino acids that turn into proteins. Primary structure is just a chain. Secondary structure is a helix or a pleated sheet. Tertiary structure is like a globular type of structure. And then the quaternary structure is many globulars put together. And then we did our activity where we learned how to use, or just refreshed our memory on how to use this chart. But in order to use this chart, you have to have the mRNA sequence done. And that's just done by base pairing. So does anybody have any questions on DNA replication, transcription, or translation? So I hope this activity today, and along with the live lesson, helped you guys understand the point of those. these concepts are right up there with photosynthesis and cellular respiration, but I think they're actually a little bit easier. So if you can understand what we talked about today, how replication works, how transcription works, how translation works, yes, I'm going to ask it on a DBA, you'll be good, 100%. So just make sure you're able to just understand and know about it, and you'll be good. So thank you guys again for coming. Next week will be the review for your upcoming DBA. So it'll be Module 3 for biology, Module 2 or 3 for anatomy. So make sure you guys come then, and I hope you guys have a great rest of your week. So thanks for coming.